Hello, dear students. Today we are going to speak about chromosomes. What is in a chromosome? A chromosome consists of hundreds or thousands of genes. A gene is the basic unit of inheritance and specialized parts that are thought to be important to the chromosome stability and function. The deoxyribonucleic acid that makes up the genes is packaged with the aid of proteins to form a complex structure. Chromosomes also contain small amounts of ribonucleic acid. DNA is packaged in chromosomes. Each human chromosome contains one very long DNA molecule, which unraveled would measure about 4.8 centimeters in length. The total length of the DNA in the nucleus of a human cell has been estimated to be about 2.2 meters. This poses a packaging problem. How does a chromosome measuring on average 6 micrometers long contain about 8,000 times its length of DNA? The answer is that chromosomal DNA is intricately folded and is tightly bound to protein molecules called histones. Histones are small proteins that are rich in the amino acids lysine and arginine. The complex formed between DNA and the histones is called chromatin. Chromatin takes up stain and is visible in non-dividing nuclei. Individual chromosomes can be seen under the light microscope only during cell division, mitosis or meiosis. Nucleosomes, the basic structural unit. Each DNA molecule is wound around histones arranged in groups of 8 known as octamers. The DNA and octamers form bead-like structures known as nucleosomes. Positively charged groups on the side chains of the histones form strong ionic bonds with negatively charged phosphate groups in the backbone of the DNA. In each nucleosome, a length of DNA containing about 150 base pairs is wrapped around the octamer. Another histone molecule attached to the outside of the nucleosome binds DNA to the octamer. The nucleosome is regarded as the basic unit of the structure. The linker region, the stretch of DNA between the nucleosomes, varies in length from 14 to over 100 base pairs. Nucleosomes fold to form solenoid fibers. More histones in the linker region help to fold the thread of DNA and nucleosomes into a tightly coiled structure called a solenoid. The solenoids are thought to be further looped and coiled around non-histone proteins called scaffolding proteins. The precise details of this higher level of folding are not known. The centromere. Each chromosome has a centromere which usually appears as a contraction when the chromosomes condense during mitosis and meiosis. The position of the centromere can be used to distinguish between different chromosomes. Centromeres do not contain any genes, however they do contain large segments of highly repetitive DNA called alpha satellite DNA. This is thought to play a significant role in centromere function. The centromere contains a kinetoka. This is a densely staining structure that attaches a chromosome to the spindle apparatus during nuclear division. Centromeres control the distribution of chromosomes during cell division. Chromosomes that do not have centromeres cannot divide. Telomeres. Telomeres are located at the ends of chromosomes. They consist of DNA and protein. The telomeres appear to play a vital role in maintaining the stability of the chromosomes, sealing the ends of linear DNA. They have been likened to the tips of shoelaces and have a similar function to stop the DNA fraying. They also seem to play an important role in regulating cell division. Under normal circumstances, telomeres become shorter and shorter with each cell division. When the telomeres have shortened to a certain critical length, the cell stops dividing. If the telomeres are removed, the chromosome disintegrates. It is thought that the aging process may be linked to the telomere damage. Telomeres, a role in cancer. Telomeres contain repeating sequences of bases which are synthesized with the help of an RNA-containing enzyme called telomerase. Telomerase activity is suppressed in normal human somatic cells. However, in cancerous cells, telomerase is active and maintains the telomere length so that the cells continue to divide. It is thought that this abnormal retention of the telomeres is involved in the development of some types of cancer. That is the end of our video session. Next time we are going to speak about inheritance. Thank you for watching.